more than 30 years of gene therapy expertise in another rare genetic disorder involving muscle, Duchenne muscular dystrophy. Then on the FOP disease side, we have Drs. Goldhammer, Kaplan, Levy, Shore, and Yu have joined this working group to provide their knowledge and insights into this research. And I just want to take one quick moment just to acknowledge the fact that we would not be having this webinar today if it was not for the work of Drs. Kaplan and Shore at UPenn back in 2006 in discovering the FOP gene. So we're very grateful for that work that has led to our ability to even conduct gene therapy research. So hopefully that provides enough context of why we're having this webinar and why now. I just want to shift gears just a little bit and take a quick journey back to your 8th grade biology class and revisit some concepts that maybe you forgot over the years, or maybe you fell asleep during that class. I'm not here to judge. So all cells in our body contain genetic material called DNA, and units of DNA are called a gene. And genes are blueprints or a recipe for building proteins in our body that help our body function. Genes also are the blueprint for many of the traits that we express, including whether you have blue or green eyes and the shape of your eye. They also help express whether you have wavy or straight hair, and whether or not you're able to run a 100-meter dash in 10 seconds or less or not. Today we have between 20,000 and 25,000 different genes, and we inherit our genes both from our mother and our father. So just to recap, a normal gene produces a normal protein that helps her body function normally. But sometimes what happens is you get a change in the DNA or a change in the gene, and this is called a mutation. If you think of DNA as spelling out a very long word, a mutation really is just a substitution of a letter for one letter for another, or sometimes it's a deletion of that letter or an addition of that letter. Mutations can come from a variety of different ways. It can come externally. Chemicals in our environment, UV can also, UV light can also cause mutations. And sometimes we inherit mutations from our mother and our father. But what happens when you get a mutation or a change in our DNA, you get proteins that may look and behave differently than the normal protein. Sometimes you also might get no protein made at all, or very little amounts of protein. And in both cases, whether you have protein that looks different or proteins that do not produce at all, you may get changes in how your body functions. And in some cases, mutations can actually result in disease. In FOP, for example, we have a mutation in the ACVR1 gene. And as a result, we get a protein that looks and functions very differently than the normal protein. And in this case, this protein sends the wrong message in our body to make more bone. So what is gene therapy? Gene therapy is an attempt to correct this mutation that results in either a protein that doesn't function correctly or results in no protein by adding or changing genetic material so that we get normal functioning protein and so that our body can function. It's really not more complicated than that at a high level. However, how this actually gets done is slightly more complicated. But in essence, that's what we're trying to do in gene therapy. Again, we're trying to get that normal protein expression occurring by adding or changing genetic material. So depending upon the mutation or depending upon the disease, Researchers use different approaches to treating that disease. And they, as Betsy mentioned quite well, there are different approaches to gene therapy. There is no one single approach. And these approaches tend to fall into one of four different groupings. There could be others, but generally these are the four big ones. We have gene silencing, gene addition, gene replacement, or gene editing. And these concepts admittedly are a little bit more for the advanced class. So what we thought we would do is walk through an analogy to try to explain the different approaches that researchers use to treating different types of mutations. So let's pretend we're all chefs and we all own our restaurant, and we're looking to make for dessert some coffee cakes. So we all go to our favorite cookbook and we open it up and we turn to coffee cake recipe. And then we read the recipe and it says add 2 cups of flour, 1 teaspoon of vanilla, 5 pounds of sugar, and 1 teaspoon of salt. Obviously, 5 pounds of sugar is not the right quantity. We have a mistake in our recipe. And as a result of this mistake, we are now producing coffee cakes that are way too sweet. So we're now going to take this analogy one step further. Let's assume we have 2 different factories making this, this cookbook. We have the blue factory, which is making the correct version of the cookbook with the correct recipe, 5 teaspoons of sugar. And then we have this red factory making the incorrect cookbook with the incorrect recipe, 5 pounds of sugar. And as a result, we're getting coffee cakes that are way too sweet. Hopefully you're with me so far. In gene silencing, we're not changing the blue factory at all. We're allowing the blue factory to continue to produce correct copies of our cookbook. However, we are going after that red factory and we're throttling it back. So only a few copies, or no copies if preferred, of the incorrect version of the cookbook is produced. And this term is called gene silencing. And in the disease world, this is a great approach when you're producing protein that's toxic to the body. So diseases like Huntington's disease would be a great way of using gene silencing where the resulting protein is toxic. Maybe FOP might be a good approach to using gene silencing. Now on gene addition, what we're doing is we don't have a blue factory necessarily, or we introduce a new blue factory to make correct copies of our recipe with that make correct versions of our coffee cakes. So gene addition is a great approach where you're producing no or very little amounts of protein, and your body just needs a little bit of that protein to be able to function. And this is a technique that might be used in conditions like hemophilia or some of the lysosomal storage disorders where you have little to no protein. Gene replacement is where you swap out factories and you take out the red factory and you put in the blue factory, and you're only making copies of the correct version of the cookbook. And then finally, gene editing, we're opening up the cookbook. We're taking our pencils with our eraser. We're changing the actual recipe itself. We're changing 5 pounds of sugar to 5 teaspoons of sugar. And this is really analogous to actually editing the actual DNA and going right to the source of the DNA to make these changes. Gene editing is definitely a newer technique that's being investigated, but it's also a pretty powerful technique out there for gene therapy. So this is the 4 approaches. Hopefully it makes a little bit more sense through our analogy. If you have other analogies, please feel free to email me. But again, I think as Betsy said quite well, the key takeaway here is there is really no single approach to gene therapy. And I know people who will be investigating FOP gene therapy will be looking at a variety of different approaches to treating FOP. Okay, so one of the important requirements for gene therapy is you need to be able to get this genetic material into the cell. The problem is that cells are really good at regulating what could get in and what can get out of cells. So what researchers have come up with is these delivery mechanisms of delivering that genetic material into a cell. Think of it almost like a letter envelope to get into your house. So it's really a unique way of getting something into a cell. But of course, researchers aren't going to call this a delivery mechanism. They're going to come up with a fancy word, and their scientific word for delivery mechanism is called vectors. And this is just a way, again, of getting something into a cell. There tends to be two different classes of vectors that researchers use in gene therapy. One is viral vectors, another is non-viral vectors. I'll just say I got to acknowledge that presenting on viruses during a pandemic may seem in poor taste. But we know all too well, I think we're all becoming viral just by just what's going on, but we do know that viruses are very good at picking the lock of cells and gaining access to cells. However, unlike the coronavirus, these viral vectors are manufactured by scientists so that they do not replicate in cells and they do not produce disease. So they do not make you sick. Their only purpose is to carry the genetic material that we want and to gain access to the cell. Once it's in the cell, the viral capsule gets cleared out. The other good advantage of viral vectors is that they are quite good at targeting specific parts of your body. So if you wanted to develop a gene therapy, for example, for your lung, but you don't want the delivery to go to your liver, say, these vectors are actually quite good at making sure that the viruses only go to, or the gene therapy only goes to the right organs. There are also non-viral vectors such as liposomes and lipid nanoparticles. These are very promising areas of gene therapy, but I would say just today a little bit more work has been done in the viral vector field, but that's not to say that these are not promising areas for further gene therapy development. So for those of you who've attended our webinar on clinical trials, this slide might look familiar to you. This basically shows how a drug goes through a process to become an approved medicine that your doctor can prescribe. I won't go through any sort of detail of this slide other than say that there are steps and processes starting in the lab, work in animals to study drugs further, and then these drugs have to be tested in clinical trials before they become an approved medicine. In order to ensure that these drugs, whether it's a gene therapy or any other type of drug, these processes are really necessary, and there are no shortcuts to ensure that the drug both works and is safe for use. And the other thing to point out is that the timelines for developing these drugs, whether it's gene therapy or any other drug, can be quite lengthy. While our gene therapy work is really starting out on the left side of your screen, the good news is we do have several promising drugs that are currently in clinical trials for FOP. So there is no doubt that gene therapy holds a lot of promise for genetic diseases. However, gene therapy, like any drug development program, has its own set of risks, challenges, and there is no guarantee that any drug that starts will ever finish. But with that said, some unique challenges exist with gene therapy. For example, first and foremost, as we discussed, we need to make sure that any gene therapy that's developed is safe and that it doesn't elicit any type of bad immune response in our patients. FOP is a disease that turns on the gene throughout a large portion of the body. So we need to make sure any gene therapy correctly targets the right cells. and that the expression levels of the genetic material that gets delivered is high enough to work. We also think of gene therapy as hopefully just one injection and we're done and that's it. But the reality is we really don't know how long these gene therapy products will last, how durable they are. However, we are learning from other diseases about this question. And then likewise, we really don't know, or we would love for these gene therapy products to reverse the disease. But today we really don't know about these gene therapy products, whether they're preventative or reversal. I personally think just from where we are today and what we don't know, it's probably safer to think of this more of a preventative approach than a reversal. But that also has to do with when you start treatment. And then I'm just going to end here with some additional resources for you in case you want to follow up and learn more about gene therapy. The American Society of Gene and Cell Therapy has a great educational page, which you can learn more. The FDA is also a good resource. It's a very high level, but this is also a good place to learn more about gene therapy. This is a long URL. If you want to get here, I would just suggest Googling FDA and what is gene therapy, and you'll get there. And then I found this other resource. It's in a slightly different field. It's in hemophilia, but Hemophilia Forward actually has some great content. It's put out by a company that developed it, but I thought a lot of the content is quite good. There's some videos, although some of it touches upon hemophilia. There is some very good background and general information. So with that, I just want to thank you very much for listening and joining Ning on your time and participating in our Q&A.
Thank you so much, Betsy and Adam. So just as a refresher, we are taking questions. You can submit them through the Q&A button that you'll find there near the bottom of your screen. So you're welcome to submit your questions there. We do have a couple of questions also. So let's start with the first question. So perhaps you could talk a little bit more about the durability of gene therapy. Is our body fighting against it? Is it reverting back to its natural state? Is it, and then where does the inserted or corrected genetic material go?
Betsy?
Okay, yeah. So I guess starting with the last part, where does the inserted or corrected genetic material go? That depends on both the genetic material that you are wanting to add and also the delivery mechanism. Different delivery mechanisms will go to different places in your body and to different cell types. And in terms of the durability, our cells naturally turn over. Our cells all have a lifespan. Some of them renew on their own, some of them live and then die. And so depending on which cell type your gene therapy is going into, those cells may continue to renew and continue to use that new genetic material to make protein almost on an indefinite basis, whereas other gene therapies may go to cells that don't renew, that have a shorter lifespan, and so then that gene therapy gets lost once those cells get lost. So that's a little bit about how the concept of durability is thought of in gene therapy.
Okay.
Anything else you want to add to that?
No, that was well said, Betsy.
So another question is, someone mentions, I've heard delivery mechanisms are a challenge in FOP because of fear to induce tissue damage. Is that correct? And are there other gene therapy challenges specific to FOP that we're aware of at this time?
Betsy, I'll take that last one. You want to take the first?
Okay.
Yeah, so as we talked about, one of the major challenges in all diseases, so this is not just specific to FOP, but it's exacerbated by FOP, is the involvement of one's immune system. One of the challenges is trying to get particles that are actually viewed by your body as foreign into your cells before your body elicit any types of immune response. In FOP, we know that any type of immune response can be exacerbated. This can be a precursor for flares and sometimes for the formation of more heterotopic ossification. Obviously, that's something we want to avoid, and that will be a priority #1 for any gene therapy before it gets developed.
And in terms of muscle, I think my understanding of the biggest challenge is simply getting a gene therapy to the muscle. For a variety of biological reasons, muscle is a harder tissue for a gene therapy to reach than, for instance, liver or an eye. So it's actually reaching the muscle that is the big challenge, and why people talk about the challenge of FOP, the challenge of gene therapy in a disease like FOP, where you need to reach the muscle to make a difference.
I just add one point on top as well, is just another overall challenge, maybe it's answering my question, is so in some diseases, we talked about where you have a mutation and you produce no protein, right? And you just need in some cases, you just need a little bit of protein. You might need 1%, 2%, 3%, 5%, 10%. That's an easier hurdle than in FOP, where we have a gene that's kind of on all the time and is throughout our body. So we need the expression, we need to turn off, all these genes throughout our body. So that's just a different additional hurdle that I think we need to consider for development of a gene therapy for FOP.
Right, so there's a question about in vivo delivery of gene therapy or ex vivo. So do we have any idea of which it might be for FOP? Or maybe it would be helpful to explain to everyone what those two types of delivery mechanisms are.
Yeah, Adam, I'll take a stab at in vivo versus ex vivo. So there are two general types of gene therapy, or two ways that you can classify them. In vivo gene therapies are gene therapies that you would administer to the body, and for instance, by an IV infusion or by an intramuscular injection. So the gene therapy goes right into your body and does its work inside your body. Ex vivo gene therapy, cells are taken out of your body, and then genetic kind of surgery is done on the cells outside your body. That new genetic material is added to the cells when the cells are outside your body, and then the cells are given back to your body, and they migrate to the place where they need to do their job. We don't know yet which type might work for FOP. That's one of the very early questions that we need to understand around the technology.
Okay, very good. So another question, let's see, that was asked is around maybe talking a little bit about, there was kind of two questions about this. We discovered the gene in 2006. Since that time, what are some of the key highlights that have happened that have led us to where we are now? And one of the resources that is available to us, and Adam, the resources that you had on your last slide, I can send that link out to all of the attendees. There is also a really great page on the IFOPA's website. It's ifopa.org, and then the word about, and then a dash, clinical trials. And it's a great place where we have a recap of all of the clinical trials that are active for FOP right now. So I know there was a couple questions about other clinical trials. So we'll provide that resource to you. So I think, Betsy, one of the questions is, if gene therapy is being studied now, should I continue to participating in current clinical trials, or should I be waiting? Talk a little bit about that.
Yes. So the road to a gene therapy is a long one. And so if you are a patient or family member wondering if this research should change what you should do, the answer is no. You should keep doing what you need to do to take care of your FOP symptoms today. You should keep taking the medications that are right for you today. And you should keep participating in the clinical trials that are right for you today. To get to a gene therapy is going to be many years of work. We have to go through the early understanding of which technology is right for us. Then we have to be able to test it in animals. Then we have to be able to think about what a clinical trial for a gene therapy for FOP might look like. That clinical trial will have eligibility criteria in order to answer the question clearly and quickly about whether gene therapy works in FOP. Trials have to have eligibility criteria, so they won't be open to every patient with FOP. And so once we can go through that whole clinical trial process, then the gene therapy might be widely available, but again, it may not be available or helpful for everybody. That's one of the many questions that has to be answered. So it's so important to stay engaged today and focus on what is available now and what might be helpful for you now. Michelle, I wanted to come back to the other question that you mentioned, which is, I think the one that gene was discovered in 2006. So sort of what's been happening since then. I would say in the world of FOP, what's been happening is understanding all of the things that happen around the gene and how that gene works and what's going wrong and how it is exerting its effects on the body. All of that biology and pathophysiology needed to be understood better. We need to understand where we can intervene in the process. And then in parallel, the field of gene therapy was evolving. And following some of those early challenges that Adam described, the whole field had to evolve and it had to continue to develop more data and for everybody to get comfortable that there was enough data and understanding to be able to safely test certain gene therapies in patients. And so that body of evidence and the understanding of the science and the science itself was all progressing. And then I would say at about 10 years ago, there were business leaders that also picked up on the science and then were able to help translate that science into something practical, into the development of therapies that could be tested in people. And so that started to generate more and more data and understanding, which now is sort of leading to a little bit of this snowball effect that you saw on Adam's slide, where now we're seeing a real uptick after all of this learning and progress and development of data. And now we're really starting to be able to test out multiple therapies for multiple conditions.
Right. Absolutely. Yeah, they all the steps build on one another. And we're finally at the place where we can take this on. That makes sense. So one of the questions that was asked is around, let's see, I had it here. Do we know if one dose, I guess will be the word that I use, one dose will change all the cells in the body, or is that something we're going to be learning in the process of the research?
Yes, you want me to?
Go, your turn.
I think the short answer is we don't know. We want this to be kind of one dose and done. There's a lot of things we just don't know in terms of, can the gene therapy get to the right cells, how much expression, and the durability, as we talked about, is just going to be an open question. And we're learning quite a bit from other fields, but until we do it and learn it in FOP, we really won't be able to answer that question. Is there anything else to add?
So another question that was raised is a little bit what I think you started to speak to just now, Adam, and it was about let me find it here. There's lots of questions coming in. So since the progenitor cells are not really known yet in FOP, which cells would you target? And is that also part of the research that we're going to be, this early research that we're going to be doing?
Yeah, and I'll go back to the that working group that we formed. Many members of that working group have such great steep knowledge of FOP and have done a lot of work in characterizing different cell types to target for gene therapy. We know a lot more today than we did just a few years ago about FOP, and I think one of the benefits of this research grant that we're funding of UMass is irrespective of whether or not it ends up you know, being successful or not, we're going to learn so much more about the cell types. And that's the thing about, I think we have to keep in mind about research in general, is that we've learned from our failures, and failure is a part of research in general. We are not always successful. It's just what do we do with the data that we have on hand, even if it's not a success. And I think the more we get into this field, the more we'll begin knowing about which cells to target, and we'll just continue learning. But again, just going back to the working group, I think that's one of the advantages of the kind of format that we've set up.
Yeah, no, that's great. That's great. There are a couple of questions that always come up in our community about people that have the variant mutation. So will we be looking, do we know if the gene therapy would have to be looked at also in the variant models as well, or, and for children, that was the other question that was raised. We had a couple of those questions around children and around people that have the variant type.
I wish we could phone a friend, because Dr. Shim, who might be with us, but I'll say this. So with regards to the variant type, I think it'll depend on the approach. We talked about those 4 different approaches. There are others as well. If you talk about like a gene editing approach where you're targeting that very specific gene of FOP, that would probably be different. Whereas if you're adding in correct versions of the FOP receptor, maybe that's enough. So I think, I hate to use the word it depends, but I think at this stage, it'll depend on the approach to gene therapy that we use. Betsy, I don't know if you have any other kind of comments.
That's great.
Let's see. There was a question here about are gene therapies for other conditions proving to be permanent, or do the effects tend to wear off? Are they seen that in other conditions?
I would say that we don't know yet. Gene therapy hasn't been around long enough for anyone to know if this is permanent. Certainly that's the goal, but we don't know yet. And time will have to pass, and more data has to be generated, and more patients have to be followed for us to know, to really know how durable these gene therapies are. And the durability may vary between different gene therapies and different diseases and different cell types, and how they work in the body.
And then someone asked the question around the research that's being done at UMass of the 4 different types of approaches that you talked about, Adam. Do we know which are being tested at UMass?
We do. We have some inclination, but I think out of respect for that research, I won't say which ones, but I will say it's multiple approaches. I think they're going to look at, they're going to really try a variety of different approaches to tackle FOP. Yeah, I'll just leave it there, but this is a great team that we have, and they're not going to just stop with one approach. That's what I can say.
That's great.
Good question, though.
So a question was asked about, scientific studies and trials are expensive. So the IFOPA has started this research grant with this campaign. So I think one thing that's important to share is that, so this campaign, the In Pursuit of a Cure campaign, really is funding 2 projects. So since 2015, the IFOPA has had a program called ACT for FOP, and that stands for Accelerating Cures and Treatments. And it's a competitive research grant program where we put out a call to researchers around the world, and researchers are invited to submit their ideas for the development of new drugs to treat FOP, new or repurposed drugs to treat FOP. And so we have a scientific advisory board that vets those various applications. And then based on the funding that's available to us, how much funding we have each year, then we award those grants. So one piece of this campaign is still focused on developing and adding new Act for FOP drug development grants this year. And then the other piece of this campaign is funding this new research grant that is focused entirely on gene therapy, the work that's being done at the University of Massachusetts. So the question is, are there other types of funding that are available besides the current campaign? And I think that, oftentimes that we see when people come to us for grant support, they may have at their university or their institution, they may have other ways of raising additional funds to help fund that research. So it's not always just the IFOPA that is responsible for funding that. So they have all of the research labs at the universities are active fundraisers too, and they are writing grants and working with donors and soliciting funds that help fund both FOP research and the other types of research they do. So, but you're right, it is expensive, but we are excited that we're at a place now where we can begin to try early gene therapy research and begin to help researchers vet what direction should gene therapy potentially head in. Okay. Let's see, what other questions?
Nicole, sorry, Michelle, maybe I'll just read Dr. Machina's comment. He left a great comment, and while you're checking for questions, we can just share that with everybody. So he wrote in that tissue damage by treatments would be a potential issue. And if therapeutic material is injected locally, it might prompt HO. And so investigation into new delivery systems might be an important success factor for FOP gene therapy, including transdermal gene delivery. And I think it's a great comment. And it speaks to how Adam described earlier that there's the genes itself that get delivered, but there's also the way that you deliver them that matters. And that's another part of the research that will be explored and another opportunity for innovation.
Absolutely. Let's see, someone asked the question, how many years will the studies take on average? And I think, I think we can say it varies from study to study, but we know that this initial study that we're funding at UMass, I believe is initially a 1-year grant, if that's correct.
Yes, the grant is 1 year, and that's again just that'll look at benchwork. That won't include any type of studies in human trials. It's ways off until we can get there. But I'll just one other general comment is one of the better points about starting now than say 5 years ago is we're allowing the kind of industry to evolve, the regulations to evolve, clinical trials to kind of evolve. So we get the advantage of seeing all this innovation in other diseases and then being able to apply it to FOP. So while things seem a little bit long now for gene therapy, I think there's a lot of lessons learned that could hopefully accelerate it in the future.
Absolutely. So a final question, I think, Betsy, that we'd like to just have you address is, what role should the community play, the FOP community play in this gene therapy research?
Yeah, it's a great question. And the IFOPA and the FOP community has a really important role to play. First, in funding research like this that lays the groundwork for gene therapy development. By funding this type of early research, we are enabling the generation of data and the de-risking of a program so that people, drug developers might be interested and able then to take the work forward. So we're really laying important groundwork by supporting research like this. The second thing that the community can do is help researchers continue to better understand the symptoms and the experience of the disease. And one of the best ways that all of you as patients and families who are here can do that is by participating in the registry. The more patients who participate and the more consistently they participate and the longer they participate in the registry, the more we are going to understand about the disease and the more we are going to enable every researcher and every drug developer to design effective studies and therapies for FOP. And lastly, I would say that just staying connected to each other, to the IFOPA, to all of the other FOP patient organizations around the world, is really important. By staying connected and in communication with each other, you are making the community stronger, and stronger, well-connected communities are more likely to get treatments. And that harkens back to the very foundation of the IFOPA by Jeannie Pieper as a pen pal organization. It is that spirit of connection that is so important, and it is as true and relevant today when we talk about this really sophisticated and innovative technology of gene therapy as it was back then, when we were just, when the community just needed to be in touch with each other through a newsletter. So, so those are some of my thoughts on what we can do as a community.
Oh, that's great. Thank you. Thank you, Betsy and Adam, for really walking us through that and responding to some of the questions. I do want to take just a moment just to remind you of the upcoming event for the In Pursuit of a Cure campaign. So I'm going to share some slides here just to quickly make sure that you have all the knowledge as this campaign continues to move forward. So it is the In Pursuit of a Cure campaign. You can find all the information about the campaign by just going to ifopa.org /cure FOP. We hope that you saw our launch video, and you can find it also on the website or on our YouTube channel to learn a little bit more about the campaign. But what we're excited is the next opportunity for you to participate in something together is the event, and it's going to be happening on August 26th and 27th. And it's In Pursuit of a Cure, an evening of inspiration and possibility. So we are going to be putting together an overview of all of the various research work that's been happening, especially focused on the Act for FOP program and the new gene therapy grant. And you'll have an opportunity at this event to sit at your home at the time that works best for you and watch this video and learn more about the Act for FOP and the gene therapy programs. And you're going to hear in this video, you're going to hear more from the researchers that are actually participating in those programs. So you'll get to hear from two of the beneficiaries of the Act for FOP grant program. You'll get a chance to hear from Dr. Gao and Dr. Shin and Dr. Shore and others on the gene therapy consortium. So we are offering it at 5 times. You'll be able to watch it either on Facebook or YouTube, and it will be broadcast 5 different times so that usually, no matter where you're living, that there will be an evening option where you'll have the opportunity to watch this. And again, if you just go to ifopa.org /cure FOP, you'll be able to access all of this information again. If you go to the Facebook page under the events, you'll find an event for the Evening of Inspiration and Possibility. You can mark that you're going, and there's a great opportunity here for you to hit share. You can share it to your Facebook page, or you can invite specific friends. But we do hope that many of you have been doing fundraising or raising awareness in your local communities, in your circles of influence, and you talk to them about the things that the IFOPA is doing, but they never really get a chance to hear from researchers or to hear from other families as well. And that's what they're going to get an opportunity to do at this event on the August 26th and 27th. So we do hope that you'll take advantage of that. We are through the program. Our goal is to raise $250,000. And again, it's going to fund the Act for FOP drug development grants and the gene therapy grant. We do have $50,000 in matching funds available. And I think there's still about $20,000 that's still available. So if you are interested in making a gift, or you know someone who would like to, if they give now, that gift goes twice as far. And you can find a way to give by going to ifopa.org /cure FOP and then _give. So again, we're just thrilled. We're grateful that the IFOPA and the community has come together since 1988, since our founding, for connection and support, and to continue to support research. And because you've been doing that for so many years, that's how we are a sophisticated enough organization now that we're able to take on a new research area like gene therapy. So it is because of you, the families, it is because of the researchers that we are able to do this now. And so we're grateful for your support, and we hope to see you at the event on August 26th and 27th. If you have any additional questions that we weren't able to answer today, you can send an email to together@ifopa.org, and we'll get back to you. But we will follow up afterwards with some of those important links. So thank you for taking time, whether it was your afternoon, evening, or morning, to be with us today. And we hope to see you on August 26th and 27th. Again, thanks, Betsy and Adam. Everyone have a great evening.
Thank you.
Thanks,
Bye.
guys. Bye.